third and final part of our chapter 6 cast of characters video will be on cellular microbes and other organisms. So you'll recall that as a cellular organism, these organisms can synthesize a cell membrane. Okay? And then the next question we're going to ask is, does this cellular organism have a nucleus? And if the answer is no, then we refer to the microbe as a prokaryote. Unicellular, one cell, lacks a nucleus. Now they do have chromosomal DNA. Most of them have a circular single chromosome made out of DNA, but there's no nuclear membrane, nor do they have other membrane-bound organelles. They lack mitochondria, they lack chloroplasts. Now from lecture you'll recall that in the three-domain system of grouping cellular organisms, two domains, domain bacteria and archaea, contain all the prokaryotes. Now, domain archaea, you'll recall, these are unicellular prokaryotes that often live in extreme environments of temperature, pH, salt. So we often refer to them as extremophiles. The second domain of prokaryotes are domain bacteria, and this is where we spend most of our time in lectures talking about numbers of domain bacteria. So in lab, we're going to be looking at some examples of bacteria. And the first one we'll be taking a look at will be Anabaina or Nostoc, N-O-S-T-O-C. Anabaina and Nostoc are what we call Cyanobacteria. They make chlorophyll A, and chlorophyll A permits them to carry out oxygenic photosynthesis. And you can see the summary reaction for oxygenic photosynthesis here. In oxygenic photosynthesis, in the presence of light and chlorophyll A, inorganic carbon dioxide and water can be used as reactants to form organic molecules such as glucose with the release of oxygen. So you'll recall that oxygenic photosynthesis had a huge impact on Earth's atmosphere. Now the Nostoc or Anabaina that we'll be looking at, they are single cells and they look like little balls, so we would call a single one a coccus, multiple cells we would call cocci. And you can see that these little cells are smaller. These are the cells that have chlorophyll A and are carrying out oxygenic photosynthesis. In contrast, you'll see occasionally in a chain a larger cell called a heterocyte. The older name is heterocyst. The heterocytes contain really cool enzyme complex called nitrogenase. And nitrogenase permits nitrogen fixation to occur. Molecular nitrogen from the air is reduced into ammonia. Now the reason this is a really important process is that most organisms can't use molecular nitrogen as a source of nitrogen to make amino acids and proteins, nucleotides, and nucleic acids. The molecular nitrogen has to be reduced to ammonia before organisms can use it as a source of building those nitrogen-containing organic molecules. So we owe a great debt of gratitude to cyanobacteria for helping to fix that nitrogen in a form that we can use. Okay, and oops, we're still within domain bacteria, so we're gonna ooze our way over here to look at a bacterial pathogen by the name of Bacillus anthracis. Now, Bacillus anthracis, as you might guess, causes the infectious disease anthrax. And there's three ways that we or other organisms can become infected with the anthrax bacterium. And most means of transmission involves this tough resistant structure called an endospore. Now, endospores are the second most resistant biological structure known. Endospores are formed to protect a copy of the bacterium's chromosomal DNA when environmental conditions are getting really, really bad. So when the actively metabolizing bacterium will potentially die because the environment is deteriorating, the bacterium can make a copy of its chromosomal DNA and then lay down layers and layers of proteins and peptidoglycan to protect that copy of DNA. So if the so-called vegetative cell or mother cell dies, that would be this rectangle here, the endospore would be released, and it can remain infectious in the environment for years, decades, maybe even centuries. So if we were to inhale the endospores would germinate, giving rise to metabolically active bacteria that would start pumping out toxins that potentially could kill us. So that would be inhalation anthrax. If endospores get into a cut in our skin, we can develop cutaneous anthrax. If we were to ingest the anthrax endospores, maybe we're eating meat from an animal that's died of anthrax, then we can develop gastrointestinal anthrax. I know it's a little bit hard to see here, but this would be a stained photomicrograph, light photomicrograph. And it's hard to see, but here the individual Bacillus anthracis cells, they look like little sausages. And we use the term bacillus for a single little sausage or cigar-shaped cell. And if we have more than one, we'd call them bacilli. And you can see that when they divide, they often remain attached to one another, forming long chains. So we call chains strepto. So in your stain smears of Bacillus anthracis in lab, you'll see that they're making these long, beautiful streptobacilli. They almost look like filaments or hairs in the microscope. All right, so our last example of a bacterial member of domain bacteria is another pathogen. Pathogen literally means something that makes us suffer. So this is Neisseria gonorrhea, responsible for the sexually transmitted infection called gonorrhea. And we'll be looking at these beautiful urethral smears from, this is not good for the patient. The patient is suffering with Neisseria gonorrhea infection. And it triggers a really strong inflammatory response with recruitment of lots and lots of leukocytes and initially lots and lots of neutrophils, also called PMNs, polymorphonuclear leukocytes, to the mucosa where the Neisseria gonorrhea are growing. So especially in men, this inflammatory response, it can create this kind of creamy white thick discharge from the tip of the urethra. So the samples we'll be looking at is that some of that urethral discharge has been smeared over a slide and then heat fixed, killed, so the bacteria are no longer alive, and then stained. And what's so lovely about those smears is you can see the little tiny Neisseria gonorrhea. Here's an electron micrograph, so we'll use this. So we see individual Neisseria gonorrhea. They have a ball shape, so we'd call a single one a coccus. Here we have two, so we'd call them cocci. And Neisseria gonorrhea, after they divide, often they won't separate, forming these pairs. So we'd call this a diplococcus. And in your smears, you'll just barely be able to see the Neisseria gonorrhea, but then you'll see these great, big, beautiful multi-lobe neutrophils or polymorphonuclear leukocytes, PMNs. They're like the first responders, the EMT, the paramedics that arrive at the scene of invasion and try to phagocytize, try to ingest and kill these bacteria. So in those smears, you're actually seeing cells of two different organisms, the prokaryote, domain bacteria, Neisseria gonorrhea, and then how would we classify the organism that made these big neutrophils? So cellular, eukaryote or prokaryote, okay, eukaryote has a nucleus, domain eukarya, and we'll see that animals, humans, belong to kingdom Animalia. Okay, so now we're going to back up here. We're going to go back up where we were discussing our cellular microbes, cellular organisms. We said no nucleus present prokaryotes. Now we're going to come back here and say, what about microbes, cells that have a nucleus? Well, we call those eukaryotes. Eu meaning true or good, and then karyote referring to the nucleus. All eukaryotes belong to domain eukarya. All eukaryotes belong to domain eukarya. And so one of the kingdoms is kingdom Plantae. And we want to remember that the beautiful Elodea leaves that we saw in Chapter 5 microscopy, you made the wet mounts of them. Those are members of kingdom Plantae. And I think over here on the side I have a model of a plant cell. And just wanted to use this to point out that here's the beautiful nucleus right here. This big structure is called the central vacuole. And what do you think these little green structures are right here? You saw them in the Elodea leaves. Through cytoplasmic streaming, they are moving around the outside of the cell. Those are the chloroplasts. So you want to remember that according to the endosymbiotic theory, the chloroplasts evolved from primitive endosymbiotic bacteria. They contain chlorophyll A, and they're responsible for oxygenic photosynthesis. Okay, now we'll go back over here to our chart. All right, and then we will next come to this kind of, this is always a challenge. You might recall that in the old days, there were lots of unicellular eukaryotic microbes that weren't plants, they weren't animals, and nobody knew exactly what to do with them. So they threw them into kind of this catch-all kingdom called Kingdom Protista. Once we had DNA and RNA sequencing available, it was discovered that members of the old Kingdom Protista, many of them were not closely related at all. So instead of Protista, I'll try to describe them as eukaryotic microbes. Most of them are unicellular, which are neither plants nor animals nor fungi. So a couple examples here. In the old days, to try to bring some order to the chaos of all these little unicellular eukaryotic microbes, some of the protists would be classified according to whether they had animal characteristics or plant-like characteristics. So the protists that had animal-like characteristics were classified as protozoa, almost like little primitive unicellular animal cells. And the reason they had animal characteristics, they lacked a cell wall. Many of them were motile, they could move. Most of them have an ingestive form of nutrition. They bring food particles, organic molecules into their cells, and then they digest them inside the cell. And the one we have here for you is a little free-living aquatic microbe called Amoeba proteus. And you can see all these beautiful little cytoplasmic membrane extensions. These are called pseudopods, false feet, and they are used by the amoeba to crawl over solid surfaces. They're also used in the process of food acquisition, the process called phagocytosis. And we've got an amoeba model here, so let me see if we can just take a peek. Here's our beautiful amoeba model, so you can see the lovely you can see the lovely pseudopods. And over here we have an example of phagocytosis. So a food particle, the pink guy, is being encircled by the pseudopods. They'll eventually fuse, forming a phagosome. And then the food will be digested within the phagosomes inside our little amoeba. Here is the nucleus, so we know it's a eukaryote. And this is a cool structure called a contractile vacuole. It helps pump excess water out of the amoeba so it won't undergo osmotic lysis. Remember, they lack a cell wall. All right. And then we're going to come back to this old group Protista, and we'll come up here to a group of unicellular eukaryotes that belong to this old kingdom Protista. But these are plant-like protists because they have chloroplasts just like plants do. And the one we'll be looking at is this beautiful organism, Spirogyra. And you'll see in your slides this beautiful helical, almost like a green spiral staircase. And that beautiful helical structure is the helical or spiral chloroplast, Spirogyra. And we'll probably also see some slides of Spirogyra undergoing sexual reproduction. It's really cool. Okay, we're going to back out. So under domain eukarya, we talked very briefly about plants. We talked about the old kingdom Protista. Now let's move to kingdom fungi. And we'll have two labs just focused on fungi, so we're just going to give you a couple of examples. So fungi, they lack chlorophyll A, so they can't carry out photosynthesis. They have to have preformed organic molecules, and there are great recyclers in nature. They help degrade dead organic material, so those elements can be reused. There's different ways of classifying fungi. Unicellular fungi are called yeasts. So you're probably familiar with the yeast, baker's or brewer's yeast, called Saccharomyces cerevisiae. It's used for making leavened bread. It's used for making wine and beer. And this is because it carries out this cool process called alcoholic fermentation, where sugars can be broken down into carbon dioxide and ethanol. And this is a way that the nutritive value of foods such as grape juice and germinated grain can be preserved. The ethanol will halt spoilage microbes from growing. So in our chart here, we can see an electron micrograph of a yeast undergoing a type of asexual reproduction called budding. So budding, instead of a single cell growing bigger and dividing into two equal parts, a cell will grow big and then it almost looks like a little nose. That's the bud. So that bud eventually is going to separate and it will grow bigger as well, and then it too can bud. So budding is a classic way of asexual reproduction in yeast. Another yeast that probably you're familiar with is an opportunistic pathogen called Candida albicans. Candida albicans can cause opportunistic infections in the mouth that we call thrush, can cause anal rectal infections, and the infamous vaginal yeast infection in women. And then another group of fungi, remember the yeast we said were unicellular. Another group of fungi are multicellular fungi that grow in long chains, and these long chains form these beautiful filaments. These are referred to as hypha, so we can see some hypha here, And the filamentous fungi we're going to take a look at is rhizopus, so-called bread mold. And we're going to have rhizopus, you'll have slides of rhizopus undergoing asexual reproduction right here. And we'll just try to see if we can't literally tease this out. So when the rhizopus is prepared to undergo asexual reproduction, there'll be a long hyphae, an aerial hyphae, that will grow upward in the air. And at the end, this big balloon-like structure called a sporangium will form. And within the sporangium, asexual spores called sporangiospores will form. And when the spores are mature, the sporangium will break open just like a balloon breaking open, and the little spores will be blown far away on air currents. Now, when environmental conditions are changing, and if we have two compatible mating types of rhizopus in the environment, the two mating types of rhizopus can grow lateral hyphae that when they meet, they'll form these cool structures called zygosporangium or zygospores. And it's within the zygosporangium that sexual spores, zygospores, will be formed, and then through a series of steps eventually will be released. So we can see the rhizopus looks really different when it's carrying out asexual reproduction or sexual reproduction. Okay, we're almost finished. We're going to come back here. We're still working on domain Eukarya, and I think our last kingdom here is kingdom Animalia. We want to remember that humans belong to kingdom Animalia. Animals are eukaryotes. They contain mitochondria. They don't have chloroplasts. I think we've got an animal model here. Here we go. Here's this great big beautiful nucleus and endoplasmic reticulum, endomembrane system. There's a cytoplasmic membrane, but there's no cell wall present. We've got these beautiful mitochondria, which recall endosymbiotic theory. We believe that the mitochondria evolved from primitive aerobic respiring endosymbiotic bacteria. Okay, so humans belong to kingdom Animalia. Insects and ticks also belong to kingdom Animalia, and blood-feeding insects like mosquitoes and blood-feeding ticks are important because they can spread pathogens between hosts. So here we have an example of Aedes mosquito. The Aedes mosquitoes, Aedes aegypti was called the yellow fever mosquito because it carried the virus that causes yellow fever. Aedes aegypti, Aedes albopictus are really important vectors in transferring Zika virus. So we know right now we actually have transmission of Zika virus within Florida, and the vectors there are these Aedes mosquitoes. Another important genus of mosquitoes are the Anopheles mosquitoes, because the Anopheles mosquitoes carry the pathogen we call Plasmodium, which causes malaria. So that's another really important arthropod vector. And one more little animal of interest over here are parasitic worms. And so we'll be looking at Taniosolium, the pork tapeworm. And there's two ways that we humans can get infected. If the pork tapeworm is growing the intestine of a human or, say, a pig, it grows in these little segments called proglottids. And the proglottids are little egg-producing factories. So as the proglottids mature, they're released from the end of the tapeworm, and then so they exit the body in feces. The proglottid dries up, the eggs are released. So if feces then contaminates food or drinking water and we consume the food or drinking water with the eggs, Then the eggs will reach our intestines, they'll hatch, and the little baby worms, the little larvae, will burrow through the intestinal wall, and then they spread throughout our body. And unfortunately, they can spread to many different tissues, including our brain. And in our brain, they'll form these little resting larval stages called cysticerci, and they can cause a lot of tissue damage there. And indeed, somebody that's been infected over and over again, they can have so much damage to the brain, a condition called neurocysticercosis, they can die. And there have been patients presenting at the UCD Med Center with neurocysticercosis, so you can see this in your career. This is a photomicrograph of the head of the tapeworm called the scolex, and you can see it has hooks with which it uses to latch onto the intestinal wall, and these great big suckers right here, so it doesn't want to get pooped out of your intestinal tract. Another way we can become infected is by eating pork, pig meat that is carrying those cysticerci. So if the pig meat, the pork, isn't cooked properly, the cysticerci won't be killed, and when we swallow the cysticerci, the cysticerci will emerge and mature into these adults that latch onto our intestines, and then we become these wonderful egg-laying machines. We can have self-inoculation if we don't clean our hands after we defecate. We can swallow the eggs that would be in the feces, and then of course we can be a source of eggs that could infect other animals. I think that does it, folks. So I hope you're really going to enjoy looking at this wonderful cast of characters.